Today on the Smart Soul Hub podcast, I want to start with something a patient said to me recently. They asked, if diabetes damages nerves by raising blood glucose, why are you injecting glucose around my nerve to fix it? Same molecule, opposite outcomes. I've been working through the mechanistic literature to find out why, and what I found surprised me. This is part 7. Hi, I'm Jae Kim, podiatrist. I hold a master's in neuroscience by research. Before I was in clinic, I was studying how nerve cells die in neurodegenerative disease and developing assays in a cancer diagnostics lab. Now I use that background to do something most podiatrists don't: go back to the primary research and ask what's actually happening at the cellular level. This is Smart Soul Hub. This is part 7 of the perineural injection therapy and dextrose evidence series. Let's get into it. Here's what surprised me. I went into this first group of mechanistic studies expecting scattered, inconclusive findings. 4 studies, completely different experimental models, different species, different cellular targets. I expected them pulling in different directions. Instead, they all pointed the same way. But before I get into what they found, let me start with what they ruled out, because I think this is actually the most important finding in the whole group. It's not osmolarity. When you inject any solution into tissue, you change the local osmotic environment. Hypertonic solutions, high concentration ones, pull water into the tissue, cause swelling, increase blood flow. That's real, and it happens with high concentration dextrose. But 5% dextrose is essentially isotonic. It sits at roughly the same concentration as your body fluids. No osmotic pull, no tissue swelling from osmosis. 2 independent experiments confirm this. In one, researchers compared 5% dextrose against fructose and saline. Both matched for osmolarity, same osmotic pressure. Fructose and saline, no pain relief. Dextrose, significant pain relief lasting over 9 days. Osmolarity is not the mechanism. Something specific about glucose is doing the work. So we're looking for something receptor-mediated, metabolic, or both. Here's the finding I keep coming back to. Some patients tell me the relief comes fast, within minutes of the injection, and I've never been fully able to explain that, because anti-inflammatory effects don't work that quickly. Something else has to be happening at the moment of injection itself. It turns out there's a receptor in peripheral nerve endings called ASIC1A that responds to the mechanical pressure of the injection volume. When the fluid goes in, the pressure activates this receptor. That triggers the release of a signaling molecule called substance P. But here's the surprising part. Substance P is textbook propane. Every physiology lecture will tell you it drives central sensitization. It's the molecule that turns up the volume on pain signals at the spinal cord. But in peripheral muscle, at the injection site, it does the opposite. It activates a different set of channels that hyperpolarize the nerve membrane, essentially quieting the nociceptors that were maintaining the chronic pain. Same molecule, opposite effect, completely context dependent. The researchers confirmed this by blocking the receptor pharmacologically and then using mice that genetically couldn't produce substance P. Both times, the pain relief disappeared completely. The pathway is real and it's specific. And critically, it only worked with dextrose, not fructose, not saline, glucose specific. This is an acute mechanism. It operates in minutes, which might explain that fast initial relief some patients describe. The second thing glucose appears to do operates over a longer time frame, hours rather than minutes. 2 studies used human neuronal cells in a lab and deliberately inflamed them with a molecule called TNF-alpha, one of the main drivers of nerve pain in entrapment neuropathy, Then they added glucose at various concentrations and watched what happened. At around 25 millimolar, which is the estimated tissue concentration after a 5% dextrose injection dilutes in the surrounding tissue, glucose switched off something called NF-kappa B. NF-kappa B is essentially the master switch for the inflammatory cascade. It controls the production of the cytokines, the inflammatory signaling proteins that sustain neuropathic pain. When glucose suppressed it, the downstream inflammatory markers dropped significantly. The cells that had been metabolically failing from the inflammatory insult recovered. There's an important caveat here though, and I want to name it because I think honesty on this is important. Nobody has actually measured what the glucose concentration is around a nerve after a subcutaneous injection and tracked it over time. The 25 millimolar figure is a calculated estimate, a reasonable one, but still an estimate. Below about 12.5 millimolar, the anti-inflammatory effect was minimal, so there's a threshold. And whether clinical injections consistently reach and maintain that threshold in living tissue is genuinely unknown. But if they do, glucose isn't just providing nutrition to a starving nerve. It's acting pharmacologically, switching off an inflammatory pathway. The third finding is the one that most surprised me, and the one I'm most cautious about. The same research group went further downstream in a follow-up study. They found that glucose at therapeutic concentrations neutralized a 40% spike in reactive oxygen species, the toxic byproducts of inflammation that damage cell membranes and DNA, returned to healthy baseline. They also found that cells which had been arrested in the cell cycle by inflammation, essentially stuck unable to divide or repair, were released from that arrest by glucose exposure. The cells could repair their DNA rather than heading towards cell death. And then there's CREB. CREB is a transcription factor, a protein that activates genes, specifically genes involved in neuronal survival, plasticity, and long-term repair. It's not a molecule that produces a quick effect. It produces lasting changes in how a cell behaves. Glucose exposure significantly upregulated CREB in these inflamed nerve cells. Now, here's why I think that might matter clinically. One of the things I observe with perineural injection therapy that I've never had a satisfying explanation for is that the relief often continues to improve after the treatment series ends. It doesn't just plateau when the injections stop, it keeps getting better. If glucose exposure is genuinely triggering CREB activation in peripheral nerve tissue, that gives us a plausible molecular explanation for that pattern. The glucose doesn't stay, but the gene expression changes it triggers. Might. I want to be clear. This is a hypothesis. CREB upregulation in a neuroblastoma cell line in a lab dish is not the same as CREB activation in a compressed nerve in a human patient, but it's a mechanistically coherent hypothesis, and it's one I hadn't encountered framed this clearly before. So when I put these findings together, three things emerge. Something acute happening at the moment of injection itself, a receptor-mediated signal that quiets nociceptors within minutes. Something operating over hours, glucose switching off the master inflammatory switch and reducing the cytokines maintaining nerve pain. And possibly something longer term, gene expression changes that outlast the glucose signal itself. These four studies were not designed together. The researchers weren't coordinating. The fact that they converge on the same directional answer from different experimental models, different species, different cellular targets is worth taking seriously. That said, the honest caveat: none of this has been confirmed in humans. The mouse fibromyalgia model is not the same as tarsal tunnel syndrome in a person. Neuroblastoma cells in a lab dish are not primary peripheral neurons. The tissue concentrations used in the lab studies are modeled estimates, not measurements from actual injections. What these studies give us is mechanistic plausibility, a framework for why perineural injection therapy might work the way it clinically appears to work. That's genuinely useful, but it's the beginning of the explanation, not the end. The full post with all the references is at jaekim.com.au/blog, link in the show notes. Thanks for listening. I'll see you next time.